me now?
Yeah.
You are hearing me now. Whee! Miracle of miracles. We've all heard of clinical trials. How many people here have been on a clinical study, clinical trial of some sort or another? I have. It's only a very small number. That's interesting. Probably reflects the number of clinical treatment studies that are going on. We hope they're going to increase. We had a very encouraging meeting this morning with colleagues from the pharmaceutical industry, all of whom are being positive about the way trials are evolving in their area. Now, being on a clinical trial is more than just passive participation in treatment. There are two key elements which you need to understand in the context of clinical studies, and those are patient-reported outcomes and the use of biomedical research behind a clinical study. They're not just part of treatment trials, they can also be used in diagnostic and care-focused studies, so don't think of clinical studies as purely treatment. Now, this context of other things that we need to think about and be aware of is what we're going to talk about in this session. Patients and patient advocates also have an important role in offering valuable insights to help shape and design a study. And unquestionably, one of the most important ways we can do this is through responding to the need for patient-reported outcomes and being aware of what patient-reported outcomes are. Everyone gets fed up with questionnaires. Another question for you. How many of you have answered a questionnaire during your clinical treatment? Oh, many, many more hands go up here. Yes. Our job as patients is actually helping researchers get the best questionnaires out. And everyone being asked to complete a questionnaire diligently is another really important step. And these are things we can get over when we talk to our patients, as patient group leaders. Now, I've recently been working on the development of a surgical study, starting from the initial question, through design, objectives, etc., etc. The study actually went into the clinic just a month ago, and it's got about 23 patients in it. It's about using a fluorescent dye in sarcoma surgery, and the chief investigator, a surgeon of course, was most interested in the surgical outcome, improving the tissue margin. And that is still the primary outcome in the study. But from the moment he talked with me, I made it clear I wanted to be able to see whether the surgery had more benefit than just that better margin. Is there a benefit in local recurrences? Will it improve the local recurrence rate? And is there a price to pay in terms of function? Loss of muscle, loss of tissue during an operation. We also need to know what that is all about for the patient. So we have good follow-up routines, and we're using the TESS questionnaire from Canada, a PRO questionnaire, patient-reported outcome questionnaire, on limb function after surgery. And we've also built into this a scientific study looking at the impact on the tumor tissue of the fluorescent dye, which helps the surgeon. Is there a longer-term impact on tumor tissue? Important information. So there we are. We've got a clinical study where we're trying to get a much more balanced view than the chief investigator started with in his ideas. A balanced view, maximizing the data we can get and ensuring that it's useful data. And patients help with that, even if it's simply just giving their approval to tests. Now, to talk about patient-reported outcomes, I'm going to welcome Tessa van Amerongen. Is that correct?
Yes.
Tessa is from Olga Hussen's group in the Netherlands, working with Olga and with Wynette van der Graaf on various aspects of PROs and quality of life. And she also leads a group at the EORTC Quality of Life group, which is one of the primary research groups in patient-reported outcomes. So Tessa, welcome, and over to you.
Yes, thank you. So, thank you everybody, I'm very pleased to be here, and I must say that entering this conference I already saw that so many people are so kind, so eager to meet each other, so that's very nice. But today I'm going to talk about the power of the patient's contribution to research, because we think that is a power and that is something fundamental in the research of the future. So, first of all, my name is Tessa van Amerongen. I'm a medical doctor at the Medical Oncology Department, but I'm also a PhD candidate in the field of health-related quality of life among all sarcoma patients and some specific research about GIST patients. And as Roger mentioned, in the team of Wynette van der Graaf and Olga Husson. So, first of all, we all agree that patients matter, but are we really measuring what matters to them? So do we really know what is most important to them? For that, we think that what is needed are patient-reported outcomes. But what are patient-reported outcomes? So PROs are direct reports from patients about their health without clinician interpretation. And this is a definition from the FDA. So it means it is directly from the patient. But we have more PROs than ever, but are we asking the right questions to the right patients? So that's what I'm going to tell you something about. First of all, PROs include a range of outcomes, so symptoms, fatigue or pain, but also functioning skills like physical functioning, social functioning, but of course also very important emotional functioning. And we also have the point of health-related quality of life. But what is health-related quality of life, actually? So here we have a broad definition, and it means like we have a multi-dimensional concept of the patient's perception of the impact of the disease, but also its treatment on physical, psychological, and social functioning. So it's quite a broad definition, and here you can see it in a figure. So, of course, we have the characteristics of the individual, the characteristics of the environment, and then the biological and physiological variables. And then we come to functional status, but also symptom status, and also the global quality of life and the satisfaction. So this is just to keep in mind as a background. Then why do we think that PROs are so important? So nowadays we're shifting from a tumor-centered evaluation to a more patient-centered evaluation. So we have for traditional endpoints like radiological response, progression-free survival, overall survival, but we also have the physician-reported adverse events. So that are adverse events that the physician thinks the patient has, like what they mentioned, for example, at the outpatient clinic. But we think that patient-reported outcomes are just as important because we want to capture the true burden of the treatment, but also enable the clinical benefits assessment, for example, of results coming out of trials. We want to improve clinical shared decision-making, like the physician can tell the patients, this is what happens with your health-related quality of life or with your functional daily life. And we also want to inform policy and drug approval to tell them what is necessary to get this new treatment on the market. What we also do from daily clinical practice, what we know is that we have benefits of these patient-reported outcomes. So several studies already stated that they demonstrated that there was an improved survival with PRO monitoring versus no PRO monitoring in routine care. And that's probably because we know better where the patient is about, where it's heading, what their symptoms are, because we don't not only ask them in the outpatient clinic, but we also heard from them in patient-reported outcomes. So we know also from research that symptomatic toxicity are frequently underreported by clinicians. But we also know that PROs are vital to detect symptoms, to monitor symptoms, and to also have an early intervention on when it's going wrong. So we need precise assessments of how patients feel and function to offer additional information and evaluating the risk and benefits of treatments. Vinetta van der Graaf was also in a team, and also Roger wrote a very nice article about how we can personalize this kind of medicine and how health-related quality of life is important, and also specifically in patients with sarcomas. But of course we want to know how and are PROs are already measured in sarcoma research. So one of my colleagues did a broad systematic review, so a literature search, and she did search in all literature to look at randomized clinical trials of systemic therapy for advanced soft tissue sarcomas, and looked, do they use patient-reported outcomes already? So at the end, she had 59 articles included, comprising 43 randomized clinical trials, and only 15 of these randomized clinical trials included PROs, and none as a primary endpoint. Then we also saw that only 10 of these 15 clinical of these RCTs reported PROs in a publication. So some did use them in their trial, but did not publicate about them. But we think they are important. So she said that recent RCTs, like later in 2015, more frequently included PROs. However, they did not report it, or they had a subject in delay in publication, or they were of poor quality. So as a result, it was difficult to compare these studies, but also difficult to implement findings, because we do not know what is the true benefit of how the patient feels after these kind of treatments. So again, are we measuring the right things? So most studies use generic questionnaires. So of course we have the European Organization for Research and Treatment of Cancer, the ERGC, and they have a very nice core cancer generic questionnaire, but it's for every cancer. So is it also specific for sarcomas? That isn't. We also have some other questionnaires that were used in randomized clinical trials, but we saw that these questionnaires did not capture disease-specific symptoms, no treatment-specific toxicities, and they also did not include contextual factors, like, for example, where the tumor was. So maybe that tool works in general oncology, but it can fail in rare diseases like sarcoma. There was also another study that used PROs, but asked, like, are they fit for the purpose? And this was in all soft tissue sarcoma patients. They identified 39 PROMs, so PROs measurements, and the mostly used was the TESS, the Toronto Extremity Salvage Score. And of course, that's not that weird, because most sarcomas are in the extremities. So the research focus was mainly on physical functioning, but there was a major gap about mental and social well-being, which is also very important. So the test is widely used for patients following surgery, for tumors in the muscles or in the bones. And there are some questions in these questionnaires that are very specific for that ones, but it is for all these kind of tumors. So Actually, nowadays we do not have a PROM that is currently recommended because we have limited evidence, we have a high risk of bias, and there are inconsistent findings in the literature. And what we also know is that the test included sarcoma patients, but that's the only questionnaire that included sarcoma patients. All of the other questionnaires were based on other patient groups. The only thing with the test is that it has a bit of a poor development quality and a weak validation. So the current PROM landscape in sarcomas is fragmented and not truly patient-centered. So we need to find something else. But then another questions pop up, and I think maybe you have the same, because can we raise standards of health-related quality of life measurements in sarcoma patients in a tumor group that is so heterogenic? Because this can be very complex. We have different subtypes, different tumor locations. We have childs, but we also have all elderly people, and there are different treatment types. So this can be hard. So is it even possible to develop one instrument that fits all patients? But we're trying. So we came up with a study in the framework of the EuroTC, so the European Organization for Cancer. And we were trying to incorporate the patient's voice in sarcoma research in how can we assess health-related quality of life in this heterogenic group. This is our publicated study protocol, and we think that patients should not only report the outcomes, but also help develop the outcomes measurements. So it's a study with the principal investigators of Olga and Winet, and I'm the coordinator of the study. And we're now at the end of phase 1, and we already had 31 centers that collaborated from all over the world. So we want to raise standards, and if it is not possible to do it in one module, maybe we have to look into a flexible approach with you have, and I will come back later to that, which you can cherry pick the items that are important for that specific tumor location or from that specific patient group. So phase 1A was already finished, and we did a broad literature search, like looked into everything. Are there already things that we know from side effects, from health-related quality of life, from everything that was reported about sarcoma patients? We did interviews with healthcare professionals, but also with patients. And at the end, we brought this all together, and then we had a comprehensive list of 386 quality of life issues that were specific for this group, but these were all issues that were just mentioned, so we were not sure if it was specific for everybody. So first we did this broad literature search, and there we saw again that there were many questionnaires were used, mostly generic, that we saw that patients with sarcoma reported lower quality of life than the general population, and that we saw distinctive patterns between health-related quality of life outcomes according to tumor location. So tumor location was very important. We also saw that in metastatic sarcoma, mostly constitutional symptoms like more fatigue, pain, but not specific symptoms were there. We saw that specifically the extremities and axial skeletal sarcomas had reduced physical functioning and disability, but for example the head and neck sarcomas had more neurological symptoms. In the retroperitoneal sarcomas there were also gastrointestinal problems, and the chest and urogenital sarcomas were not even investigated yet. So we had to do something about that. Then another German study that also looked into sarcoma survivors and included more than 1,000 patients also saw that health-related quality of life significantly differed from tumor location, and that many treatment-specific issues were not captured in these core generic tools. It's a bit small, I don't know if you can see it, but here were some results from that study, and the left part for you is the functional skills, and that means how higher the bar is, how better. And the right side are the symptom skills, and that is how higher the bar is, how worse the symptoms are. So at the left side, you can see that the axial skeleton sarcomas have actually the most worst quality of life and also worse functioning, and also have the highest symptoms. But for example, skin sarcomas has less symptoms and higher functioning skills. So they are very different from each other. There was one other study also that looked into quality of life from advanced sarcoma patients that received first-line chemotherapy. And it was a prospective study. They actually also saw something that we have to keep in mind, is that the global health status declines at all patients over time, and the decline was observed regardless of response. So if they had progressive disease, stable disease, or partial response, their health-related quality of life declined. And the main driver was treatment toxicity and not only disease progression. So we see sometimes that chemotherapy has a modest survival benefit, but the toxicity burden is substantial. So this study also shows that without PROs, we risk overestimating benefit and underestimating harm. Then again, this German study, I think I already had it maybe, so I'll move on. So with our phase 1A study, we looked into all these interviews and did a qualitative analysis. So we searched for every word that was in the interviews and tried to put it in domains. So we looked into these two groups because we saw they were understudied. And to have a bigger view on this group of the breast sarcoma patients and the thoracic, we did at the end mapped it like this. So looked into the domains of physical health, mental health, and social health, and tried to get everything out of these interviews. Out of these, we at the end got all these issues specified, and then they were at the end getting in our comprehensive list of health-related quality of life issues. And here you can see the burden that these patients had. So all this research shows us that there is a big heterogeneity of the disease, and specifically on tumor location. So one instrument may be challenging. So maybe we need a more flexible approach, but because we had these static paper questionnaires that just followed question by question, but we also now have like more maybe dynamic questionnaires that questions are presented in a changing sequence. So if you answer this question yes, then another question comes, then when you answer no. And especially nowadays with AI that is coming, we can make it more specific to the patients. We are also using the ER2C library. This is the library of the ER2C that puts all the questionnaires that they have into one library, and then all questions that are there make it into items. So at the end we have 2,075 questions, and they are in different languages, and people can use those questions. So for example, if you want to do research about a sarcoma subpopulation, you can use the C30 to look into comparisons between other studies, but we also need to cherry pick items from the item library to look in what is the aim of the study, but also in what sarcoma subpopulation do we investigate. So now we are analyzing the results of our phase 1B, and that is that we use this comprehensive issue list of 386 issues. We asked, but also the C30 and location-specific modules, for example, bone metastasis or breast cancer module or the head and neck cancer module. We asked all those patients to score this issue on relevance. Is this a relevant issue for you? And they could score 1 like not relevant, 2 a little relevant, 3 relevant, and 4 very relevant. And they did score everything. In the end, we had 536 patients that scored it, and also 85 healthcare professionals that scored it. And now we're analyzing, like, are there issues that are for the broader sarcoma group? And we mean with that that it must be in 5 out of 10 tumor locations, but also 2 out of 3 treatment groups. But we also are looking now at, are there like specific issues for specific tumor locations? For example, difficulty with a long walk, or difficulty jumping, or more gastrointestinal symptoms, so it can be everything. Then we hope to determine a strategy, for example, the C30, one sarcoma module, and an item list that is, for example, based on tumor location. So with this, we hope to, at the end, inform physicians, but also research strategies to how we measure things that are really important to the patients. But we have also one ERTC study that is more based on GIST patients, because this study is specific for sarcomas, and of course GIST is also sarcoma, but we excluded GIST because they have a different approach. But we also have this ERTC GIST study that's about the long-term survivorship challenges of advanced and metastatic GIST patients that are responding to TKIs. It's an observational study at first, because we have these patients with metastatic GIST, and we know they can live for years now, and we're so happy about that. They can live on many years on TKI treatment, but we are now shifting from a chronic treatment to a chronic toxicity, but we're not sure what are the long-term side effects of having this treatment. We do know that stopping treatment can lead to progression, and that maybe reintroduction can restore control. But we also want to know the long-term patient's experience on how they feel after such long years being still patients receiving treatment. So the aim is to look into the quality of life problems, the associated factors, but also to identify key survivorship issues and long-term toxicities. And we do that with a questionnaire that is developed with patients and very based on symptoms that are treatment-specific symptoms. So I think people that have experience with imatinib know some symptoms that are in this list. For example, swelling around the eyes, but also pain in the muscles, pain in your hands. Your eyes can be dry or more watery. And with this list, we can have more specific. We do specifically more know what patients are feeling because they defined these questionnaires. So coming to an end, we know that PROs are essential, but current approaches are insufficient. So we see that health-related quality of life can be severely impaired. It is heterogenic. The tumor response is not always similar to patient benefits, and that we have an over-reliance on generic tools, but we do not know if these generic tools capture everything that the patient feels. So we think we need to go to a more flexible, patient-centered approach to improve clinical trials, but also to improve shared decision-making, value-based care, and we need all patients to co-create these patient-reported outcomes. So it's not optional, but it's fundamental, and we need to go to a more flexible, patient-driven approach so that it will not be a long questionnaire, but just specific for your needs. And we are very curious if you have any comments or recommendations or any questions. So thank you.
Yes, I think that we're going to go on and present our next and then have a discussion all together.
Yes.
So please have a seat. Thank you very much for that. Laying the groundwork for that really important component of how patients can contribute to our research. Now we're going to turn our attention to the power of patients' contribution from the perspective of research biospecimens. And I want to begin with, this discussion actually began at our meeting last year in Washington, D.C. in the bone sarcoma track, where Patrick Rohar presented about a positive clinical trial in Ewing sarcoma. And it was a study that had shown on the on the left-hand side, you see what's a waterfall plot, and every one of the bars going down, that's a good thing. It's showing that there's a reduction in the size of the tumor. And what was particularly important, he told us, was that the people that went on to this study had 1 to 7 previous types of treatment with a median of 4, and they saw a confirmed objective response rate of 33% compared with a 10% response rate in the past. And then on the right, also importantly, you see what we call a swimmer's plot. It kind of looks like swimmers swimming in a lane, and the further off to the right is the longer that the person has been on the treatment. So in addition to having a high objective response rate, they had sustained responses. And this particular study was just recently published in Nature Medicine, so it was a very important finding and a very important step forward for Ewing sarcoma. But he also pointed out to us lessons that we can learn from samples that are merely the size of a strand of hair. So in his lab, he studies, so Dr. Grohar is a pediatric oncologist, but also a basic researcher. And in his lab, he has created cell line models of Ewing sarcoma, as well as mouse models that model the disease and that he can test different therapies in. And he had tested the therapy and got the hypothesis for doing this study by his work in the lab. So he wanted to go back and say, did we really affect the EWS fly, which is really a important hallmark and driver of Ewing sarcoma? So you don't need to be a scientist. I'm not a scientist, but if we look at the picture on the left, this is a piece of tissue that was taken from somebody no bigger than the strand of a hair, and the colored parts on there show active EWS fly. So this is the person before they had any treatment. Then they get a dose of the treatment, and then the person had another biopsy done. And you can see that there is much less of the colored cells there, showing that there is, only after one treatment, we had good number of cells that responded. There were some remaining. So he pointed out to us that this data is absolutely critical to the success of this trial and future trials. And even though the amount of tissue was really probably not as much as he would have liked, it was still transformative. So he really pointed out to us that really power in partnering together, that correlative biology is really going to be the road to progress, from this kind of sense of what did we learn at the bench, what did he see in his lab, what did we take to the clinic and do in the clinical trial, and now what are we learning from the clinical trial to go back and better understand the disease so we do even better treatments going forward. So in this particular study, out of the 38 patients that participated, 12 had agreed and were able to have biopsies. And this is really a big improvement, believe it or not. I mean, one might think that's not 12 out of 38, not that great. However, it really showed a big improvement over 15 years. And Dr. Grohar's perspective was it was really because of advocacy. It was because of patient and patient advocacy input to get those samples. It was all voluntary on the part of the patients. And while the samples, as he said, were not as large as he had hoped, they were still transformative. So I felt it was so important to continue to partner with advocates and patients, that we really need to learn from every patient, and that we need larger biopsy specimens, and we need to make sure that these important studies are funded. And so to continue this discussion, we're pleased to have Christina Iptoma and Nicholas Harold to present to us from the perspective of patient advocacy, as well as clinician and researcher, to talk about not only the need, but the challenges and the opportunities that we have by having research biospecimens. So I'll begin with you, Christina, please. Oh, okay. We'll begin with Nicholas.
Yeah, I'm so pleased to have been asked to, together with Christina, give a perspective from I'm a pediatric oncologist. I'm doing basic research, but I'm also a clinical trialist, and have this dialogue. Is it the research question that is important, or is it the patient that is important? And I think we would all agree the patient is important, but how can we sort of align these two together? Now I have to see whether it's really supposed to be me in terms of the slides. Do you want me to? Yeah. I wanted to mention something that is today's the 1st of May, and that is the International Workers' Day. I'm not going to put this into politics now, but it's about getting together and working together. So this is a good omen, I think, for the whole day, or for the whole conference. Sarcomas, they are rare, but they are not only rare, they are heterogeneous. So depending on how you classify, you have more than 100 or 150 different diagnoses. I think Felix will let us know that if you look under the microscope, this is also different from if you do genetic analysis, etc. And that is a real issue, because it's not like breast cancer, where you have, even at in a small country like Sweden, well, we are a large country, but we have a small population. There you can get hundreds and thousands of patients, but sarcoma, we have like maybe 400 diagnoses per year in Sweden, and then we have all these different subtypes, so it's really difficult to run meaningful clinical trials, at least in the conventional way of running those trials, where you need high number of patients, you give them treatment A and compare to treatment B, and then you draw your conclusions. It's rare, it's heterogeneous. It's not only heterogeneous that there are many diagnoses, but it's also heterogeneous within a diagnosis, and it's also very heterogeneous within an individual patient. If you think of osteosarcoma, not one cell looks the same genetically as the cell next to each other. And then it's even more complex if you look at metastatic sites, etc. And then you induce changes with treatments, that just like we saw from Patrick Rojas' studies. So it's difficult. So what do we need if we have a rare disease? We have to learn as much as we can from each and individual patient. We just don't have the luxury to choose. We should really strive to learn as much as we can from each patient, for each patient, and for future patients. And that means we need to collect data, and that means we need to collect biospecimens. And since many of the sarcomas we deal with, they are systemic diseases. So yes, they manifest at one site specifically, but it's a systemic disease that will spread or has already spread in many cases. So you need to understand the system, the body. With chemotherapy, we treat the whole patient, but the cancer is also affecting the whole body. What is the immune composition? Can you learn something from blood analysis? What does the tumor look like at site A, prior to treatment, following treatment, at relapse, etc. So what we show here, longitudinal. So it's not enough to just look at one point in time. In order to learn properly, you need to check what is happening to the patient and what is happening to the patient's tumor. And I want to illustrate this with why are we doing that? But the why is also, imagine one patient with the same diagnosis of, let's say, and the picture was actually taken from a leukemia patient, but anyway, say osteosarcoma or Ewing sarcoma or rhabdomyosarcoma, and they received the exact same standardized treatment, and one of the patient to the left, survives, makes it, the other patient dies, and we just don't understand why. They have the same disease, so to speak, but apparently they don't. So we need to understand, why does this treatment work for the one patient and not for the other? Another question of why is, we pediatric oncologists, we are so proud of what has happened during the second half of the 20th century in terms of survival. This is Swedish data where you see different diagnoses and different color codes, 5-year overall survival, and then depending on when you've been diagnosed, at which year, what were your chances of being alive 5 years later on? And you see most of them, they go up very steeply. So there has been a lot of improvement with introducing systemic combination chemotherapy for most part. But you also see, and here I just indicate with these thick lines what has happened during the last 20, 30, 40 years for soft tissue sarcomas, which is mainly represented by rhabdomyosarcoma in the pediatric population, and in Ewing and osteosarcoma. For the soft tissue sarcomas, basically nothing has changed in terms of being alive after 5 years. In Ewing and osteosarcoma, not so much either. I mean, there is some improvement with this condensed intensive induction chemotherapy that probably explains why there is a slight shift upwards. But this is not enough. And we're talking about 70% survival, so that means 3 out of 10 will die. And it also means here you're lumping together all the good prognostic patients. I mean, if you have a localized, easily resectable disease, this is different from a multimetastatic patient. So there are decades of stagnation, and what has been successful in the past, just trying to test. I mean, if no patient survives, whatever you throw at them will hopefully have some effect. But here we have tried for so long, and we don't see improvement. So we need to do some more. Is it now that you take over, Christina?
Oh no, you still have a couple more.
Yeah.
I think these are yours. Yeah.
And then what is important is, okay, if you do a clinical trial, you include patients, and then you have an outcome, and hopefully you learn something for future patients. We don't have the luxury with those patients. We also need to offer the patients that contribute with the tissue, with the blood, to give them some real-time benefit. And that means we should strive to do those type of modern analysis that we now have the possibility to do ex vivo drug testing, do the sequencing in a real-time manner so that we can learn and feedback for the individual patient within a clinical trial or outside of a clinical trial. But then what is important for research, of course, is the systematic analysis that we learn, that we collect from ideally all patients, to make a more systematic gain of knowledge for future patients. But it's not enough. If this is the only thing, I don't think it's ethical. We really should strive to be able to provide something already for the individual patient. And that's not just, okay, this is required for you to get access to this experimental treatment. It should really be something. We don't really know. Your odds are bad. We'll try to look under, like you say in Swedish, each rock. Is there something that we might be able to test? So, as for myself as a medical professional, the patient comes first, the patient comes second, and the patient comes third. And this requires also that we share data and knowledge. I mean, we have a career, and you want to advance both in the hospital and scientifically. But it's not about fostering your own career. You must not be protective of your own data, because after all, it is not your own data. And this is very easily said, more easily said than done, because there are incitements with funding agencies, competition, etc., that this can be forgotten. But in particular in sarcoma and in pediatric oncology, there is no way around this. Is it now you think? So, Christina, please.
Thank you. All right, thank you so much, Denise and Nicholas, for, I think, really kind of laying the groundwork for why patient biospecimens are so important. So I am Christina. I'm the Director of Scientific Programs at MIB Agents, which is a US-based osteosarcoma nonprofit. And I came into this world because my son Dylan passed away from osteosarcoma in 2021. And so I thought I would review challenges and opportunities from a patient perspective, and also maybe share some personal vignettes and stories to maybe kind of bring some of this to life. So first challenge, limited tissue from biopsies, and I think we covered this a little bit, but biopsies, especially if they're core biopsies, there's generally not a lot of tissue to deal with beyond what's used for diagnosis. And particularly, a lot of patients, when they're first diagnosed, they're potentially at a smaller community hospital. Sorry, the sound keeps, I'm going to try to move my mic a little bit. They're potentially at a smaller community hospital where there might not be any research protocol to be saving or collecting that tissue. And so, again, beyond what's used for diagnosis, that tissue doesn't usually go anywhere. So as Nicholas mentioned, it's so critical to have longitudinal data, but oftentimes that kind of treatment-naive biopsy sample we just don't have because of just logistically where it is in time. Processing and preservation, just a couple things to note here. So processing, there's still some challenges, I think, with certain disease types. I know in bone cancers, there are challenges with decalcification and processing that tissue. And sometimes there are harsher solutions that are used, and that can destroy the nucleic acid, making it really difficult to do any deeper sequencing some of these tissue samples. So there are some gentler decals out there, and I think people are using that more commonly, but it's still not universal. So that's still an issue that, we just have some tissue samples that are just not of great quality. Also for frozen samples, so cryopreserved samples are really important to do, again, like more deeper analysis. But there are definitely logistical issues with cryopreservation. So if you are at an institution where there is a steady protocol and they are planning on collecting that tissue, that will probably happen no problem. But if you, for example, want to send your tissue to another institution for another study, that requires a lot of upfront planning and careful coordination and communication and a lot of micromanaging along the way. So I'll also share kind of a vignette here. So when Dylan was going through treatment, we wanted to collect tissue to send actually to an outside commercial company to do some functional drug testing. And that required a lot of communication from us, from the patient family to our care team, and then particularly to the surgeon, who would be the one in the OR actually taking that tissue. So we had to really know that up front, coordinate with the care team, coordinate with, who's receiving the tissue on the other end. And then, everything to bringing that collection sample kit to the surgeon and making sure that everyone that we talked to, during pre-op that was going to be in that OR, they knew that this is what was needed to happen, that we wanted that tissue to get to this other third party. And you also have to have a surgeon who's willing to do that for you, because I've heard stories where some surgeons are not really open to that. Luckily, ours was, and was really great about coordinating with pathology and making sure he ran down there, coming right out of the OR. So definitely some logistical issues with getting samples to cryopreserve. Okay, consent and ethical constraints. So 3 points I wanted to make here. So consent is such an important part, right, of laying the foundation for all things to come, for collecting those biospecimens. And, there's definitely best practices, I think, for how the consent is done. And it's really important. And again, I wanted to share a couple of personal vignettes. So I remember being consented for a few studies while Dylan was going through treatment. 2 of them were in the first couple weeks probably of diagnosis, where, as many of you know, everything's a fog, right? And you have no idea what's going on. But what I do remember is being consented for a study, and I think he was probably a resident maybe. Someone must have sent into our room pre-op, and couldn't quite explain the study. And I remember I had a lot of questions because, here we are like 2 weeks into it. And he couldn't really answer any of those questions. And I think at the end of it, we probably still ended up consenting, but it wasn't very confidence building. And nor did I leave with a great feeling about participating in that study. And honestly, I still don't remember what that study was about. The second time I was consented, it was in recovery and post-op. So Dylan had just come out of the OR. We're in the room, the nurse is there. We're carefully monitoring him to make sure he has no pain issues or nausea as he's coming out. And someone runs into the room and tries to consent me for a study. And that was an example of not the right time or place to do a consent. So I know they sound really basic, but this happened. And I also understand that normally in a hospital, it's like schedules are crazy, right? There's a lot of stuff going on. But I would just say that there's a lot of opportunity to be really thoughtful about how the consent is done, both in making sure that there's someone who's really involved with the study, the PI, or if not the PI, someone else who really understands the study and can explain it to the patient well, and also at the right time and place. And then the third thing I wanted to, second thing about the whole consent and ethical constraints, research biopsies. So I think this has been kind of a hot topic lately, the idea of doing research biopsies during a study that may not have clinical benefit to the patient, but are important because of all the things that Nicholas mentioned, that we really need to understand how the tumor evolves or how it's responding to therapy. And so I think there are a couple of institutions I know in the US that are really thinking about baking research biopsies into any studies that they do moving forward. I think Memorial Sloan Kettering is doing this. St. Jude is thinking about doing this. And so MIB agents actually partnered with Memorial Sloan Kettering to do kind of a focus group with patients. This was actually a group of AYA patients to ask them how they would feel about doing research biopsy on clinical study, knowing that it may not have any direct clinical benefit to them. And I would say about, we had maybe like 8 or 10, and about 75 to 80% said, yes, of course, absolutely. I would definitely contribute samples to help other patients. And so there's definitely patient appetite to do that. But I'm going to sprinkle a little bit of salt on that, because as someone who is the caregiver of a pretty young pediatric patient, and being in that moment, because I will also be a little cynical about it and say it's probably a little easier in hindsight to go back and be altruistic about it and say, yeah, of course, I would do that. But I think in the moment as a caregiver, again, of a pediatric patient, my role as a parent was to really protect him. And he was already undergoing so much from surgeries and chemotherapy that I'm pretty sure that if someone had presented the option to us to have another procedure done that may not clinically benefit him or that was not clinically necessary, but he might have pain or wound complications or other things, we probably would have said no, just to be totally honest, in that time. So I think there is a little bit of, there are definitely some of those issues at play on research biopsies. And then lastly, on the consent and ethical thing, the other thing is just patient advocacy, agency over their own tissue. So in the US, I know it's like this, where even if the patient has their tissue, taken out at their institution, the institution owns that tissue technically. And if you have that tissue directed else to another institution or even a commercial company, the person on the receiving end then technically owns that tissue. And so I know as patients, we think of that as like, well, that came out of our body or my loved one's body, like I think that belongs to us. But technically it does belong to the institution. And so there again is this balance, which I think Nicholas addressed so nicely of, the patient might have priorities on what they want to do with that tissue. They might want to have it sent to get functional testing done at another institution or commercial company, but there are definitely also some research priorities at that institution competing, right? There are a lot of things competing for that very small piece of very valuable tissue. So it's again kind of how we balance this to make sure that the patient also has some agency over how their own tissue is used. And then Nicholas also kind of addressed the siloed institutions about sharing data and tissue. And I know that the sarcoma community, though, is a highly collaborative community, and that there are many investigators who generously share their tissue and data despite their institutional protocols around doing that. So just switching over to opportunities. So benefit to patient. I think, we do really want to align those. So there's the certainly, we can learn a lot for these samples to further research, but if we can try to align that where there's a benefit to the patient, whether or not they get information that might inform their personal treatment plan, and they may, maybe there is no information that they get from that, but there's a possibility that they might, then I think there's a greater incentive for patients to participate and contribute samples. Talked about the education and a thoughtful consent process, but I would just kind of encourage providers to really think about it as an educational opportunity to really kind of establish to the patient why it's so important to collect these samples. Sharing samples. So there are a couple of great examples on sharing. So I know also from like a patient and funder perspective, we've funded Dr. Sweet Cordero, who has a very active osteosarcoma lab at the University of California, San Francisco. And that funding helped fund different PDX and cell line development. And his lab has shared that with 20 plus institutions across the world. And so I will say as like a patient advocate funder, it's really fulfilling to know that there's been such tremendous impact from that funding investment. Also, there's another initiative at Memorial Sloan Kettering started by Dr. Filimon de la Cruz called Proxy. I don't know if maybe some of you have heard of it, but it's basically a PDX consortium. And so in addition to Memorial Sloan, there are 5 other institutions, and it's a brilliant idea. They actually now have a umbrella or blanket MTA or mutual transfer agreement to be able to share samples with each other very easily and directly without having to do a contract every single time they do that. So now if a patient comes in through, let's say, Dana-Farber in Boston, that sample can be shared with 6 institutions in this consortium. So I think we just need more initiatives like that to enhance sharing. And then lastly, sharing findings with patients. So I think it's really important, again, as a patient, you're contributing whatever it is, your blood, tumor tissue, saliva even. I think we've participated in a study where we sent in saliva. But I will say that for some of the ones that we've participated in, I don't even know if they ever even received it. I don't think I even got any notification that, yes, we received your sample. And certainly didn't really hear about how it was used or if it led to any learning. And so I think sharing information back to the patient community about all those things is really important. So I'll share another example. There's a study, mainly US-based, called Count Me In, that has focused on different diseases, and they have also done one on osteosarcoma. And their model is direct to patient. So their model is we're going to engage patients directly and then ask for the patients to consent to share their medical records and their tissue with us. It's actually part of an NIH overall study called the, I can never remember what all the letters stand for, but it starts with patient engagement. So it's actually a consortium across different institutions, and Count Me In is one of those studies. And so it's really important to them. In fact, a lot of the research in that overall effort focuses on how they communicate to patients. And so it's been really important for them to have a dashboard, for example, so that when a patient submits or enrolls, they get a dashboard tracking, yes, we received your consent. Yes, we received the medical records from your hospital. Yes, we received your tissue. And so the analysis is still going on for that study, but a big part of that study will be how do we communicate those results back to patients so they know what their tissue helped move forward. So I think just really important to kind of share the results with the patient community to really create that culture that it's a partnership between the patients and the scientific community in moving that research forward. And it also just creates a virtuous cycle. So I think knowing that, yes, my sample contributed to learning just creates that culture of being excited to continue partnering in that way. So thank you.
Right, I think that brings us to the point where we need to break for coffee. What, 2 questions then, Catherine? What do you think? 2 questions, right, 2 questions. Anyone want to?
Sorry.
If you could just please say who you are as well. Thank you.
Brandy Felser from SFA. I have a question about PROs, just like a clarifying question. I was recently at a workshop with the FDA, and we had a conversation about PROs. And one of the questions that we talked about was the fact that there's no real, exactly what you presented on, no real standard of questionnaires that are effective for sarcoma. And one thing they mentioned is that there's all these questions to choose from. So I loved your presentation because it was fantastic it just related to somewhere I was at a couple weeks ago. But anyway, the question I have, though, is when they said there's thousands of questions you can choose from, my understanding is that it has to be a validated, evidence-based questionnaire, not a pool of questions that we can just sort of pick from and then develop the questionnaire. So I'm trying to understand why he would say that, but also, you know, we've talked a lot in the patient community about getting a validated questionnaire and one that is regulatory grade, for example. And so with the comment from the FDA, and he was actually the person in charge of PROs at the FDA, with him saying that, you know, you can pick from a pool of questions, I guess I'm really interested in, with the work that you're doing, how are we getting this validated for regulatory grade? And can you clarify the fact that we can't just, are you thinking we can pull questions and create our own questionnaire? I guess I'm just trying to understand it a little better. I know I'm not feeling well. My question's not, I'm all over the place. But you get what I'm saying. Thank you.
Are you on? Have a go, Tessa.
Hello?
Yeah.
Thank you. I did understand your question, I think. So I think what is important to know about the study I presented, that it is just a phase 1 study. So we, at the ERTC, we have very specific module development guidelines to come to a validated questionnaire. So actually there are 4 phases to have a validated questionnaire. So if we ending this study, we still have to go through the phase 2 where we look into all these issues, and then phase 3 to translate it correctly and also ask another group of patients to validate it, and then we also still have a phase 4. So we're not yet there, but the thing is that we saw that these tumor locations are so important, for example. So we actually need to go into every tumor location and make a measurement strategy for every tumor location, for example. So maybe we can have a core module for sarcoma broad, but then we also have to validate everything for these specific groups and then connect issues to those groups, and then you can pick the issues that are belonging to that group at the end, because it's just not possible to have one that has it all for all sarcoma patients. So I hope that I answered your question with that. So we're not there yet, but we are hoping to get there.
So what do you think patients can do and patient advocacy groups now to help us get there?
So I do think also, it's just very important that we still have talk with each other, what is important for you when are in your chemotherapy, for example, or after surgery, or so is it more treatment specific? Is it more indeed location specific? So I think we still need those conversations with each other, and we just need all the patients to validate everything that we do. So if we have at the end this issue list that we think that is relevant, we still go again back to the patients and ask if it is relevant for them. So in that thing, I think still that they have to be part of our research group. So we are including them during our process. And I think at the ERTC there are many researchers and many project groups that are currently trying to see how patients can be more involved in these research groups and at the development and that kind of stuff. Yeah, so
Thank you.
quite extensive.
But I think it's fair to add to that the regulators are well aware of this situation. So that is not what they're saying is not an argument not to use the standard of PROs that's available now. You use them because they know the pathway that to getting to the full regulatory requirement, the regulatory rigor, if you like, is underway, and it's not going to happen instantly.
No,
Yeah.
unfortunately.
So. Speak again. Say again. Gotcha.
Yeah, if I could jump in on this discussion, there is a validated instrument that measures quality of life. By the way, I'm Jeff Kramer with the Chondrous Sarcoma Foundation from the United States. There's a validated instrument called the PROMIS scales, and they do measure quality of life. And we actually used it in our patient registry. It's interesting, you know, because they've got different measurements in there in terms of quality of life. They look at cancer patients as a whole, as a general whole, and then they compared us to cancer patients. And we found out that our particular community fares worse, you know, and what that motivated us to do was to develop support groups. And there were other issues that came out of that PROMIS scale. Patients are motivated because of pain, okay? And that seems to be number one on their case, and it came out of that instrument. And the other thing that came out of that instrument was that they're not being heard. They'll go to their doctor and they'll say, I'm suffering, you know, with a certain pain, or, you know, I'm having trouble with this and that. And it's like they're being overlooked, and it frustrates them and gets them very upset and agitated. So there is an instrument out there that does that. We need to improve on those instruments, obviously, and that goes to the discussion that you've had. But I just wanted to make you aware of that.
No, thank you, Jeff. That's valuable input. Let's move on. Yes, sir.
Yeah.
Right, just shout and we'll try and repeat.
Sorry.
Oh, well done, Catherine.
Thank you, Catherine. Yay, it works. I'll do karaoke now. Okay, this is a quick one. It's for Christine and for the panel generally, and it's a sensitive subject. I was in a workshop quite recently talking about patient samples in the post-mortem settings, so taking samples from deceased patients. And it was a subject on which there were lots of questions but not many answers. So maybe this is just to posit the question and we think about it, but if there's any advice that the panel can give when I go back to this workshop and we talk about it again, about how to encourage family involvement in giving these samples and addressing this sensitive subject, then it'd be welcome. Thank you.
Do we have time? Thank you for bringing that. Is my mic on? Can you guys hear me? Okay, great. Thank you for bringing that up, because I meant to touch on that and didn't quite get around to it. But yes, because that's actually incredibly valuable as well. And I know in the U.S. there had been an organization that was really it was like a legacy donation, because it is an incredible legacy to pass on. But the logistics, again, are really hard because there's a very small window. And so you really need to kind of think of it beforehand. And I'm sure, as many of you can imagine, it's a hard thing to think about beforehand. So I know, I've actually talked to some folks in the U.S. about this, and And actually, Dr. Delacruz at Memorial Sloan Kettering, who's the one that's put together this proxy consortium, is also actually thinking about similarly having kind of like a network where there might be distributed centers around the country where if someone was interested in a legacy donation, it's really easy for them to just like coordinate with one person and then make it happen, and then everything else gets taken care of. But it's not an easy solution, I think, to just addressing how do you speak to patients and caregivers about it, because it's a really sensitive topic. And I've thought about it personally, myself. I think no one had presented that idea to us, and I don't think it was something that was top of mind for me at the time. And I have to, again, be honest and say that even if it were presented as an option to me at the time, it was not something I think, and which is, because again, I'm a huge proponent of research, but at the time, it was not something that I could mentally wrap my head around, knowing, again, everything he had gone through, and it was just like, yep, one more procedure. And so I think mentally it's difficult. So I agree with you. It's a difficult, sensitive topic, and aside from just putting it out there and as an option for people that are interested in doing that, and oftentimes it's the patient, especially even pediatric patients or young adults who actually say, this is something that I want to do, and will tell their family or their caregiver that this is something I want to do, so can you please make sure this happens? So I guess as best as we can, just make that available and provide easy options for families to do that.
If I can just comment on that, I think this is sensitive, but it's also wise and important to consider. But what I do think we need to do as professionals a better job in is, at least, of course you want to collect samples as well for doing something that you don't know yet you want to do, but you need to give clear perspective. What can be done already today? What is the knowledge that you might gain from it, or at least with maybe you have some study that you think this will be done with the tissue in addition to other projects that could be done later on, so that you really see, okay, this is a purpose, something tangible that you're contributing with, not just, okay, they'll make holes in my beloved one just to store samples for some obscure project that might come up in the future.
Well, I want to thank
So-
Tessa and Nicholas and Christina. Oh, they were marked?
Yeah.
Okay, sorry.
Yeah. Hello, all. This is Sandeep Kumar from India. I am also Ewing sarcoma survivor. So adding to Brandy and Jeff. So my point here is that what if I am a patient and I am going to OPD and doctor's team like saying that you need to enroll for this research or study. So what I'm going to get from this study, number one, like any current benefit for me, like my file will be always on top whenever I next visit to the hospital, or will I get the like concession medicine or for the treatment care? And also my last point is that when a research or study is completed, so each patient those are involved should know about the impact, because I stand as a underwent for a research study during my treatment. The research name was TOS, and I don't know what happened post that. So it's been around 19 years of my treatment. So yeah, thank you.
You raise a good point about making sure that we communicate back what we find and how important it is. And I think that as we begin to see patients and patient advocates being more engaged in research, we can also be part of that communication of results and that we take that on as partners, recognizing that that is so important. And so Tessa and Nicholas and Christina, thank you so much for kind of sharing with us the needs from a standpoint of patient-reported outcomes and the importance of biospecimens, some of the opportunities that they will give us in the future, and recognizing that we still have some challenges ahead of us. And so as you leave the conference, we're hoping that you'll leave with some concrete ideas of things that you can do in your own communities and advocacy groups, recognizing that your voice is incredibly important for us to make progress in these areas and improve our understanding of the biology of these diseases. We can't understand it without these important biospecimens, but it's the challenge of the consenting, and we want our voice heard in clinical trials, so we need to work with you to be able to help validate those tools. So thank you all for staying over our very first session. We appreciate it. And now we'll have a coffee break. And when do we return? When would you like us back?
2:45, like 20 minutes.
About 20 minutes. Thank you very much.